Okay, I'm back. So cancer and cell growth. Let me just make sure. This was just showing you, I just went back one to show you that cancer cells do not exhibit anchorage dependence or density. Sorry.
Come on.
Harper wanted to be in the camera again. That's okay though. We're just moving forward. Or density dependent inhibition. So when you look at your lab has you look at 4 different slides of cells. And basically, when you look at these slides, what you're looking for is evidence that the cells exhibit cancer-like characteristics, okay? You are not expected to be histologists or pathologist is what a histologist looks at what normal tissue looks at. A pathologist looks at what abnormal tissue looks at. When I was in vet school, I took an entire semester of just histology. What do normal cells look like? So this lab is really just to kind of let you see how much you don't know, in all honesty. But if you look here, if you look at this cancer, this is at a very large view, but the cancer cells are on top of each other. They're quite nasty. I would encourage you to watch the video links that I included that are the cancer cells basically exhibiting normal cell growth and then the rate of cell division when it is a cancer cell. It's kind of a nasty video if the volume's on because you start hearing it crackle. Could you please shut the refrigerator? Okay. So we're going to keep going here. Cancer cells, and the reason why cancer is so brilliant at evading all of our different ways to try to eradicate it is because it's essentially a failure to control the very thing that we need to exist, the cell cycle. So our inability to stop cell division here is what basically causes cancer. It gets into the very fabric of what our cells need to survive. So unrestrained or uncontrolled growth is really the definition of cancer cells. And what control is lost? Well, we lose those checkpoints. And a lot of times it's in the coding of the genes. It might be the CDK is faulty. It might be the cyclins are wrong. One thing that we found that all cancers have in common is this gene, P53. And yes, that is in red and in bold. It's on your test. P53, this gene, seems to play a key role in all G1S restriction points. So basically, any time that we're going from G1 to S, and we said that was a real critical point, all cancers have shut down the P53 activity. So what that's saying is that cancers make it so that that restriction point doesn't exist. And a cancer cell will just keep going in this cell cycle, and there's no stopping it. Cancers, what are the main characteristics of cancer cells? These are the characteristics that you're going to be looking for in your lab. These are the things that you want to pretend that you see or you don't see on these slides. Whether or not you actually see that, it's really a needle in a haystack, okay? This lab is more for you to think about the characteristics of cells that have cancer and the characteristics of cells that are not cancerous. So basically, cancer cells do not develop as normal cells. They're going to have unlimited growth. The promoter genes, the growth promoter genes, are always turned on. They ignore checkpoints. The problem with that is those checkpoints might say, hey, all the chromosomes aren't attached, or hey, this cell should probably go into programmed cell death. It's not big enough to divide. So you start getting cancer cells start looking really funny. They're misshapen. They have extra chromosomes or less chromosomes or sister chromatids are still attached. They all turn off that tumor suppressor gene, that P53. Cancer cells are able to escape apoptosis, which is programmed cell death. So that's that suicide gene. By being able to escape apoptosis, cancer cells can reproduce for essentially ever. Now that you have all this extra time on your hands, and I might make it an assignment, would be to at least watch that movie, Henrietta Lacks. The book is actually very well done. But Henrietta Lacks, her cells, the HeLac cells, are really instrumental on a lot of our scientific discoveries. She died, gosh, I don't want to misquote it, but a long time ago, maybe 50 years ago. Her cells are all over the world and thriving because that's what cancer cells do. They don't die. So when they pulled, oops, sorry about that. We're just going to get you back up. When they pulled the cells out of her, they basically they're still living and growing and thriving. But it is definitely a really good read. I would highly, and it's on HBO. A lot of times I used to play it. I'll try to find a free link to it, but it is the Henrietta Lacks movie. Oprah Winfrey plays it. This right here is pretty phenomenal. This shows a cancer cell and all the blood vessels that are going to it. Immortality means that they have unlimited cell divisions. They also have this phenomenal way of promoting blood vessel growth to them. Okay. And they also are able to overcome the anchor dependency and density dependent. So the touch sensor gene is what we call it. This shows you a benign tumor. This is something I always just used to question when I was your age. What's the difference between a benign tumor and a malignant tumor? A benign tumor means that, you see how it's encased in a capsule? So benign tumors, a lot of times they say, oh, it's not that bad, it's benign. Sometimes they claim they're not going to remove them because it's got a capsule around it. So the idea is that this guy can't go and spread to other areas. A malignant tumor, on the other hand, leaves the original site. It'll go into the blood and the lymph system, and it starts more tumors. When we say it starts more tumors, the word that doctors will use is metastasize. The problem with tumors is that they impair the functions of the organs, that they're, whatever that organ is supposed to be doing, it can't do. The tumor interferes. This talks about treatments for cancer. There's a whole bunch, but basically the ones that we are still using and are kind of archaic, hopefully we can move forward, is all chemotherapy and radiation does is they target rapidly dividing cells. So that's why people will lose their hair. Because if your hair is a rapidly dividing cells, they end up with a lot of skin issues because skin is rapidly dividing. And what else is also rapidly dividing? Cancer cells. Oh, it's okay. You can come and sit by me. Harper's going to sit by us now. She's having a hard time with not having attention. You're doing so good, though. This is an example of a new miracle type drug that we're trying to get. And the problem with these drugs is it takes forever to get them through. Gleevec has been around for a long time. It's just not, I think it's might be FDA approved, I'm not sure. But what it does is it targets the proteins that are only found in the cancer cells. And this is cool because what Gleevec is doing that the other treatments aren't doing is the other treatments are just, they're targeting all rapidly dividing cells, which really has a toll on your body. However, Gleevec actually targets a protein that is only associated with the cancer cells, which means that only the cancer cells will die from this treatment. Okay, so that's it. I will post another video that kind of summarizes your assignment. All right, you guys have been absolutely amazing and patient in this process. Thank you so very much. Would anybody like to say, Harper, Tristan, would you like to say goodbye? Okay, say bye-bye.
Bye.
Harper? Bye. Okay. And VB? VB is asleep, but say goodbye, VB. VB says goodbye.